Good afternoon to all the students in this course. Hi, my name is Ashutosh and I'll be the TA for this problem solving session. So as we have proceeded with the TA sessions of this course, so today we'll be solving practice problem set corresponding to lectures of week 9 where Professor Subramaniam is teaching us about plant development. So we'll learn some aspects of plant development by solving these questions. And you must have also received the quiz and assignment grading. So we'll solve similar kind of problems in this practice problem session and understand the basic concepts about plan development and the right answers. So let's start. Let's wait for 5 more minutes since people are still joining. I'll keep everyone who is requesting, I'll keep admitting them into this call, everyone who is requesting to join this call right now. And I'll start sharing my screen at 3:05 and we can begin the session. So this will be an interactive session between the TA and the students. I'll post a set of 10 questions and you guys have to tell me the answers or whatever you think is the correct answer in the chat box. All communication with me will be via the chat box. And a general set of rules that I have been asking all the students to follow, that please turn off your mic and video chat during this call session. You can interact with me via the chat box. And if you have any query, you can raise your hand. I'll allow you to speak when I have finished this session. Okay, with that much set of general instructions, let's wait for 5 more minutes and I'll keep admitting students who are trying to join this call. And we'll begin sharply at 3:05. Thank you. It's already 5 minutes past 3, and we will start the session now. I'll just quickly share my screen, and please let me know if my audio is clearly audible to you and if you can see the screen. I'll just share my screen, just a minute. We do not have many students in this session, but it's fine since I'll be recording this session. I'll discuss all those 10 questions and people can refer to it in the future and clear the doubts. Okay, I hope you can see my screen and see the slide changing as well as hear my voice clearly. Just let me know in the chat box with a yes or no, then we'll proceed. Okay, so I welcome you all again to NPTEL Introduction to Developmental Biology. This is the TA course. This is the course which we will be solving problems of, and the instructor is Professor Subramaniam. My name is Ashutosh and I am from IIT Kanpur. So the format would be same that we have been solving in the previous sessions as well. We'll have a set of 10 questions similar to what we get, 10 to 12 questions similar to what we get in our assignments, and I'll pose those multiple choice questions. There can be one or multiple answers correct. We all will try to answer them based on our understanding and our memory of the subject. Then you just let me know what do you think is the correct answer. It's okay if your answer is correct or wrong. I'll reveal the correct answer and then we'll move on to a brief explanation about why the correct answer is the correct answer. Okay. So the first question that we have here, choose the correct options. You have to choose which of these is the correct option. Option A, Hox genes are arranged in a collinear manner on the chromosome. Option B, Hox genes are scattered across many different chromosomes. Option C, expression of the Hox genes is constant throughout embryogenesis. And option D, the Hox genes specifying anterior and posterior parts of the body will be adjacent to each other on the chromosome. These are the four statements and you have to tell me what do you think is the correct option among all these four statements. So read through all these statements carefully and let me know what is your answer in the chat box. Let's wait for a minute to collect some answers. Okay, it's time for revealing what is the correct answer to this question. The correct answer to this question is option A. Hox genes are arranged in a collinear manner on the chromosome. Now let us see the explanation of this answer. So here is a diagrammatic representation of Hox gene clusters, and we show a comparison between a Drosophila and a human being. So the correct answer was Hox genes are arranged collinearly on a chromosome. So the arrangement pattern of Hox genes determines their expression pattern, and Hox genes are involved mostly in anterior to posterior axis patterning. So the expression of different Hox genes at different levels along the anterior posterior axis determines different regions, different patterns in the fly body as well as the human body. And we can see here These colors denote different genes. ABDA, ABDB, UBX, ANTPs. These are Hox genes of the antennapedia and bithorax complex present in the fly, and corresponding segment or the body plan which is forming due to the expression of the Hox gene is represented here. Similarly, we can see collinear arrangement of Hox genes. Hox genes which are expressing in the posterior stay towards the end of the posterior, and then adjacently there are mid segmental Hox genes and then anterior Hox genes. So we have a linear arrangement like this, and Hox genes can be on different chromosomes, but in a single chromosome they are arranged collinearly. And also about one of those statements, Hox genes are not expressed uniformly throughout embryogenesis. So throughout the embryogenesis, not all Hox genes are expressed uniformly. Some Hox genes, so we'll see in the next slide how Hox genes are having two different kind of control, spatial and temporal control. But what happens in the real scenario instead? Their expression is tightly regulated both in space and time to ensure the proper development of embryo and the formation of distinct body structures. So this is what I was talking about. So both in space and in time. Space means, let's say in the body plan, we have anterior to posterior, so that means space regulation. Some Hox genes which are in the anterior will only express in the anterior. They will not express in the posterior. So spatial regulation is maintained, and temporal regulation, timely regulation. Certain Hox genes express earlier to specify certain structures, and other Hox genes come later in the embryonic development to specify other structures. So let us see a quick example of spatial and temporal collinear regulation. Spatial regulation is can be explained beautifully by anterior to posterior morphological gradient of expression. Suppose we have a morphogen uh, which is now diffusing from anterior to posterior, coded by a Hox gene. Such morphogen, if the will have a higher gradient at anterior if it is coded by an anterior Hox gene, and be lesser in gradient towards the posterior, determining the variety of structures along the anterior posterior axis. And temporal collinearity, early development to late development, different Hox gene activate in different manner. So in combination, temporal and spatial collinearity lead to expression of a concept called posterior prevalence, a term coined by Du Bois. This is to say more posterior genes that are expressed after more anterior ones overrule the medial or anterior class genes, in the sense that their body patterning influence is superimposed upon and in fact could be said to dominate the anterior ones. So this concept of spatial and temporal linearity or adjacentness of the Hox genes leads to what is known as posterior prevalence. Like the posterior genes, posterior prevalence we must have seen in Drosophila, where the posterior genes such as ultrabithorax overrule the medial or anterior class genes, can inhibit antennapedia. So antennapedia is the anterior gene which is inhibited by the ultrabithorax. So in absence of ultrabithorax, antennapedia can express everywhere, but due to the presence of ultrabithorax, we have posterior prevalence. The posterior segments do not express antenna. I hope the explanation is clear to the students. Now moving on to the next question. In the next question, we have match the following column, left and right. So you have to match A, B, C, D to 1, 2, 3, 4. Option A, on the match column A, we have hunchback and crupple, Hox gene activation, ABDB, Hox gene expression maintenance, cadherin, transcription factor, trithorax, polycomb, cell adhesion. Now what you guys have to do is match the following correct one. You match column A, B, C, D to 1, 2, 3, 4, and here are the options: A1, B3, C4, D2, A2, B4, C3, D1, A2, B3, C2, D1, A1, B3, C4 and D2. So read the question carefully on the screen, and whatever you think is the best match between A, B, C, D and column 1, 2, 3, 4, tick that option and let me know in the chat box. Let's take a minute to wait for students to answer. Okay, it's time to reveal the correct answer. The correct answer to this match the following question is option A, where A matches 1 hunchback crupple Hawk gene activation, B matches 3 ABDB transcription factor, C matches 4 cell adhesion, Catherine and cell adhesion, and D matches 2 trithorax polycomb matches with Hawk gene expression maintenance. So this is the correct answer to the question. Now let us learn about all these matches. So the first match we have is hunchback and crupple with Hawk gene activation. So hunchback and crupple are gap genes and they act as transcription factors for Hawk gene activation. Transcription factors encoded by gap genes cooperate to create even more complex patterns of gene expression. Here is an example how transcription of different gap genes, transcription factors encoded by different gap genes establish Hawk's gene expression. Expression domain of hunchback mentioned in red here, and expression domain of crupple. And you can see a fine line of overlap. The expression domains of hunchback and crupple overlap over a fine line. And in this yellow area, some genes require both hunchback and crupple present to be turned on. And just like this, if you remember, we learnt about the hierarchy of gene activation in Drosophila. First, on the top level of hierarchy, we have maternal effect genes. Then we have gap genes. Gap genes control pair rule genes. Pair rule genes control segment polarity genes, and all these three, gap genes, pair rule genes and segment polarity genes, in turn influence the expression pattern of Hox genes. So the first match, option A, would be with 1, where hunchbacks and crupple act code for transcription factors or act as transcription factors for Hox gene activation. Then for the next set of matches, where we saw the match between polycomb repressor and trithorax group in Hox gene maintenance. So PCG here refers to polycomb group of genes collectively behave as genetic repressors of Hox gene. So wherever we have polycomb group, polycomb repressor protein, here as we see here, PRC1, PRC2, we usually see a genetic repression of Hox genes. Whereas on the opposite hand, we have trithorax group of genes which are necessary for Hox gene expression or function. So we have two players here. One player, polycomb repressor, which represses Hox gene activation, and the other player we have is trithorax, which activates the Hox gene activation. So collectively, these proteins or genes maintain the expression of Hox genes, whether it is activation or repression. That's why we have matched polycomb repressor with Hox gene maintenance. and Catherine is responsible for cell addition. It is pretty straightforward. And the other one, ABDB is a Hox gene, which is a transcription factor. Yes, ABDB, abdominal B, is present in the bithorax complex. I hope this is clear to the students. And with that, we move on to the third question. Which of the following statements are true? So we have a true-false situation here. Which of the following statements are true? Option A, plants and mammals both show tremendous developmental plasticity. Option B, meiosis in plants produce spores and not gametes. Option C, multicellularity evolved independently in plants and animals. And option D, plant germ cells are determined and set aside during early development. So out of these statements, which of these statements are true? Read through all the statements very carefully and please let me know in the chat box which of these statements do you think are true. Mind you, there might be more than one multiple choice correct question answer. Let's take a minute to gather some answers and students can refer to the question. Okay, it's time for me to reveal the correct answer to this question. So the first statement that is true is option B. Meiosis in plants produces spores and not gametes. So gametes are not a characteristic of plant. Meiosis in plant germline or plant megaspore produces spores, while gametes is a characteristic of animal cells. Then the next statement, which is true, is option C. Multicellularity evolved independently in plants and in animals. So the unicellular plant, we can say that unicellular plants are algae, or we have different kinds of algae, while the multicellularity in plants developed independently. We have algae, we have bryophytes, ferns, and gymnosperms, angiosperms. These are the different evolutionary classification depending on the the style of multicellularity and body plan. While in animals we have unicellular uh, unicellular organisms like sea yeast and paramecium, and we have protists. So and then on we have higher organisms. So these two statements are the true statements. Option B and option C are the correct answer to this question. So what refers to developmental plasticity? Developmental plasticity refers to capacity of an organism to alter its development in response to environmental cues or change in its surrounding. So if an organism can alter its development response to the environmental signals, then it is called highly developmental plastic. So this is on the basis of first statement, plants and mammals both show tremendous developmental plasticity. What we would see here is plants have higher developmental plasticity than mammals. That's why the statement 1 is not correct. Plants have generally higher developmental plasticity. Depending on the depending on the conditions, it can alter its development. A small example here would be the dormancy in seeds. So seeds are already fertilized, but they remain dormant. So they trigger their developmental program active only when a certain set of conditions are met. Let's say when there is enough moisture, enough food, and ample surroundings or environment to the seed, only then the further development of the plant will begin. But that is not how it works in mammals. Once the zygote is formed, then the developmental program is triggered, and for humans we have 9 months of gestation period in which the whole development will complete and the offspring will be out. But that is not the case in plants. This is a very simple example of seed dormancy. Only when ample conditions are met, the seed will germinate into a plant. Also, multicellularity evolved independently in plants and animals, as I was saying, and plant germ cells are not set aside early in development. So, unlike animal cells or unlike animals, where we have a quite distinct group of cells which will form the germline, committed to form the germline, there is nothing such as a committed group of cells in plants which get set aside early in development. Plant have higher probability that any, so we will see when we go deeper into the plant development that when megaspore develops, it's a chance event that any of the cell can become a megaspore, and but by a chance event one of the cells survive and the other three or four die. That's how the surviving cell becomes a megaspore. And these are the plant growth and developmental stages. So the first stage, three stages of plant growth and development. The first stage we have embryogenesis, fertilization to seed. We have the embryogenesis after fertilization till seeding effect. Then vegetative growth, where we have the growth of shoot and root. Juvenile stage, germination to adult. Phase change marks transition. And reproductive development, making flowers and reproducing sexually. So these are three different growth phases of any plant, and we have just seen that plants have higher developmental plasticity, multicellularity has evolved independently in plants, and plants germline is not set aside in early development. Plants are more plastic than mammals. That is why we have these two options which are correct. Meiosis in plant produces spores. Plants go through a sporophytic phase and a gametophytic phase where meiosis produces spores. Now spores can form the gametophyte, and gametophyte will give rise to gametes. And multicellularly evolved independently in plants and animals. So these are the correct answer to this question, and I hope it is clear to the students. Okay, now moving on to the next question, question number 4. It's a fill in the blanks kind of question. The dashed stage in plants is known as the sporophytic stage, which undergoes dash to produce spores. On the other hand, the spores can undergo dash to form the gametophyte, which then produces gametes. So this is what I was talking about in the previous question. Now you have to fill in the blanks and join the dots to find the answer. Dash stage in plants is known as sporophyte stage, which undergoes dash to produce spores. On the other hand, the spores can undergo dash to form the gametophyte, which then produces gametes. And your options are diploid, mitosis, haploid, meiosis. Option 2, haploid, mitosis, diploid, meiosis. Option 3, diploid, meiosis, haploid, mitosis. And option 4, haploid, meiosis, diploid, and mitosis. So these are your four options. Read through the statement very carefully and let me know what you think is the correct answer. Put your answers in the chat box. I'll be waiting for a minute to gather some answers. Then I'll reveal the correct answer. Okay, it's time for me to reveal what is the correct answer. The correct answer to this question, this fill in the blanks question, is option C, option 3, diploid, meiosis, haploid, mitosis. So these four words would fit the statement rightly. The diploid stage in plants is known as the sporophyte stage. So always we have to remember diploid stage is sporophyte which undergoes meiosis to produce spores. So this answer was kind of hinting in the previous question. Meiosis of the sporophyte gives rise to spores and not gametes. On the other hand, the spores can undergo meiosis to form the haploid gametophyte, which then produces gametes. So there is a technical mistake here, but it's counterintuitive. Spores can undergo mitosis to form the haploid gametophyte. So these two blanks would be exchanged: diploid, meiosis, mitosis, and haploid. Then the answer becomes correct. Diploid stage in plant is a sporophyte which undergoes meiosis to produce spores. Then spores can undergo mitosis to form the haploid gametophyte, which produce gametes. So here is a cartoon representation of the life cycle of a plant with both sporophytic and gametophytic phases. We have a gametophyte N which gives rise to gamete N of diploidy with mitosis, and another gamete fuses, two gametes fuse in fertilization to give the zygote 2N. N, N, we get 2N. Zygote can undergo mitosis to form the main plant body called the sporophyte, where we have diploid. Multicellular diploid is the sporophyte, as we saw in the last answer. Then this multicellular diploid sporophyte can undergo meiosis to give haploid spores. So we have N haploid spores. Haploid spores can undergo mitosis, increase in number. So we have to remember it that meiosis is reduction division in which the set of chromosomes is halved, and then mitosis is the multiplication of cells when we have the chromosome number similar and we have more number of cells. So haploid spores undergo mitosis to give rise to gametophytic body, multicellular haploid. Gametophytes can and then undergo mitosis to give gametes, and this process can repeat again. So the 2N or diploid multicellular body is called sporophyte, which undergo meiosis, whereas the haploid multicellular is called the gametophyte, which undergoes mitosis. This was the whole answer and statement about. And this kind of life cycle of a plant is called haplodiplontic life cycle, because if we divide the life cycle of a plant, we can easily distinguish between a diploid part of its life and haploid part of its life. The same plant goes through both the phases in its life. First, it forms the diploid sporophytic body. Then it undergoes transformation into the gametophytic body via meiosis, which produces the haploid generation. So we have after fertilization, we have mitosis, 2N organism, this is the sporophyte. Path D with meiosis forms the 1N organism. So this is the spore here. 1N organism is the gametophyte. Gametophyte can produce mitosis, can undergo mitosis and give rise to two different gametes, the male and female gamete, which can then fertilize again to enter the diploid generation. So we have half of it in the diploid generation, half of it in the haploid generation. So this kind of life cycle is called haplodiplontic life cycle of the plant. Now, as we see many classification of plants, many plants have different uh, phases of life dedicated to diploid and haploid generation. In some plants like bryophyte, the haploid generation is more in time as compared to the diploid generation. The bryophytes spend most of their life in haploid generation. And while we move up in the ladder, when we reach to angiosperms and gymnosperms, the haploid generation is fairly limited and most of the life is spent in the diploid generation. But nevertheless, all plants follow this haplodiplontic life cycle. I hope this is easy and clear to the students to understand. Just let me know in the chat box. Yes, great. Okay, moving on to the next question, next set of questions. This is also a fill in the blank of a single statement, and we have question 5, 6, 7, all the three questions here, and the answers would be integers from 0 to 9. So it's a number, it's a numerical answer. The largest megaspore in angiosperms undergoes dash mitotic divisions to generate dash cells and dash nuclei. So this is what you guys have to answer. You guys have to recall the megaspore division, how many mitotic divisions it undergo, the largest megaspore in angiosperm, and what number of cells does it generate, and what number of nuclei. These are the three questions, question number 5, 6, and 7. How many mitotic divisions is question number 5. How many cells is question number 6. And how many nuclei is the question number 7. Take your time, read through the question carefully, and try to recall the megaspore division in angiosperms leading to female gamete formation. Then you will be able to answer this. Let's take a minute or two to collect some answers. Then later on I'll reveal the correct answer and we'll proceed with the explanation of the correct answer. Okay, it's now time for the revelation of correct answers. So the first answer, correct answer, the largest megaspore in angiosperms undergoes dashed mitotic divisions. The correct answer is 3 mitotic divisions. Largest megaspore in angiosperms undergoes 3 mitotic divisions, right? And generates how many cells? The next question. Correct answer to that question is it after 3 mitotic divisions it generates 7 cells. Mind you, 7 cells. It's not an even number as we expect for mitotic divisions, and we'll see why. It undergoes 3 mitotic divisions to generate 7 cells, and how many nuclei? 8 nuclei. So 3 mitotic divisions leads to 7 cells formation and 8 nuclei. That's why an egg or a megaspore is also known as 7-celled 8 nucleate staged egg. So at that stage we will find 7 cells and 8 nuclei which are a result of only 3 mitotic divisions. Now let us follow this diagram to see. This is a megaspore mother cell which is diploid. It undergoes meiosis to give 4 haploid megaspores. So one megaspore mother cell, meiosis, 4 daughter cells with half of the copy. So we have 2n to n. Out of those 4 haploid megaspores, 3 of them degenerate. and only one of them, megaspore or embryo sac, only one of them survives and grows bigger in size. Growth nourished by the nucleus. So this nucleus now nourishes the growth of this megaspore, surviving megaspore, where it undergoes the first mitosis. End nearest the micropyle, it undergoes the first mitosis. The first mitotic division happens with nuclear division, where we have karyokinesis but without cytokinesis. So in the same cytoplasm, we have two nucleus. Then the second mitosis, each of these single nucleus here divides into two within the same cytoplasm. So no cellular division or cytoplasmic division, but only nuclear division, karyokinesis. So we have four such cells now. Then we have the third mitosis, which produces this 8-cell structure. We have this division of 3 antipodal cells, 2 synergids and an egg cell. So one of these will become an egg cell, and 2 synergids for supporting antipodal cells, and 2 polar nuclei. So we have 2 polar nuclei in the center, antipodal cell, 3 antipodal cells and 2 synergids. So we have the micropylar end here and the chalazal end here. This is the root where the pollen tube will come and fertilize the egg. And we have these polar nuclei, which are joint nucleus. So we have 2 nuclei here, but 7-celled stage, because we have 3 antipodal cells, 1 egg cell, 2 synergids, so 3 plus 3 6, and 1 single cell with 2 nucleus called polar nuclei, and this whole cell. So we have 3 plus 3 6 plus 1 whole cell, 7-cell stage but 8 nucleus, and this is formed by 3 rounds of mitosis. And this arrangement is ready for nuclear fusion happens later when 2 polar nuclei fuse. Mature embryo sac, female gametophyte, just before fertilization, 7 nuclei present, 6 are haploid, 1 is diploid. So just before fertilization, if you observe, the 8 nuclei stage becomes 7 nuclei. 6 from the 3 from the antipodal, 2 from the synergids, 1 from the egg cell, and these 2 polar nuclei fuse and form one single nucleus just before fertilization, forming the 7 nuclei stage. So this is the developmental scenario of a megaspore mother cell into the egg or female gamete. I hope this is clear to the students and pretty easy to understand. You might have to go back and revise a little bit and see these diagrams and life cycles so that you will brush it up. Okay, moving on to the next question. Which of the following statements are false respect to plant development? So you have to identify the false statements with respect to plant development. Option A, self-incompatibility maximizes the genetic diversity of offspring. Option B, gametophytic self-incompatibility depends on genotype of the haploid pollen grain. Option C, sporophytic self-incompatibility depends on genotype of the diploid plant producing the pollen grains. And option D, successful receptor ligand SRK SCR binding leads to pollen tube germination in brassica. These are the 4 statements, and you guys have to tell me which of these statements are false statements, okay, with respect to plant development. Take your time, read through the questions well, and let me know your answers in the chat box. Let's wait for a minute or two for all the guys to answer, and then we'll move ahead. Okay, it's time for me to reveal the correct answer. Which of the following statements are false with respect to plant development? The correct answer is option 4. Successful receptor ligand SRK-SCR binding leads to pollen tube germination in brassica. This statement is false. While it is reverse, successful receptor ligand interaction inhibits pollen tube germination. So if you mark, all these statements are about self-incompatibility and pollen germination on the female reproductive organ, the style. So we'll understand the concept of self-incompatibility in this question. So self-incompatibility is defined as inability to produce zygotes after self-pollination in a fertile hermaphrodite plant, which has stamens and pistils in the same flower. So this is a generalized definition, pinpointed scientific definition, telling us that if in a hermaphrodite plant which has both male and female reproductive parts, that is a that plant has high chances of self for pollination, while that plant exhibits a phenomenon called self incompatibility, in which the own zygote, the own pollen from the same plant or with similar genetic makeup is rejected for fertilizing with the egg cell and create a zygote. So self pollinated is prevented via this phenomenon called self incompatibility. This structural organization of the hermaphrodite flower increases the risk of self pollination, leading to low genetic diversity. So hermaphrodite, this statement is talking about hermaphrodite. In a hermaphrodite plant, the organization risks the self pollination, leading to low genetic diversity. So the the plant has developed self incompatibility to increase the genetic versatility of the plant. Otherwise, self pollination will cause inbreeding and accumulation of harmful and recessive mutations. So to ensure that variability among the population, genetic diversity is maintained, the plant has come up with self incompatibility. Now, self incompatibility can be of two types: gametophytic self incompatibility and sporophytic self incompatibility. In gametophytic self incompatibility, in gametophytic self incompatibility, the the genotype of the gamete that is produced determines the compatibility over the pistil. So whatever gamete genotype pollen, so the pollen that we have here has S1 genotype or S2 genotype, any kind of pollen which is not S3 S4 will be able to germinate. But if it is a S1 S4 recipient plant and we have S1 kind of pollen landing on the stigma, then it won't form the pollen tube. Also in an S1 S2 plant, the S1 neither S1 or S2 pollen will form the pollen tube. So this is gametophytic self incompatibility, where the incompatibility depends on the genotype of the gamete or the pollen, while sporophytic self incompatibility is determined by the genotype of the pollen donor. So the sporophytic plant, which is the source of this pollen, that determines the incompatibility. So as we know, the exine and intine or the outer layers of the pollen are secreted by the sporophyte. So those will have the similar genetic makeup of the sporophytic plant. They will be S1 S2 for a plant pollen donor of S1 S2. Now these will be rejected in any recipient having either S1 or S2 or both S1 S2, and only when both of them are absent, S3 S4, where there is no S1 S2, only then the pollen will form the pollen tube. So this is an example of sporophytic self incompatibility. So we learned about gametophytic self incompatibility, sporophytic self incompatibility, and that self incompatibility is a mechanism to increase genetic diversity among plants. So all these both these statements are true. Gametophytic self incompatibility depends on the genotype of the haploid pollen grain. So genotype of the gamete or pollen grain, then gametophytic self incompatibility. Sporophytic self incompatibility depends on genotype of the diploid plant which produces the pollen grains. So the source plant which has produced the pollen grain, genotype of that is dependent. If it is dependent on that, then it is called as sporophytic self incompatibility. Now what happens when a pollen lands on a stigma? If we have self pollen, we have the SCR protein or SCR locus. If an incompatible mating, self incompatile, in an incompatible mating, if the SRK allele on the stigma recognizes the SCR allele from the same plant, SCR is the S locus receptor and SRK is the protein. So if the cognate receptor and protein will recognize only if they are from the same alleles. So S locus has different alleles. So what happens when tight recognition and binding happens? A signaling cascade is triggered that leads to inhibition of pollen tube growth. This prevents cell fertilization. So actually the signaling works to inhibit the growth of pollen tube when the same kind of or self incompatible pollen arrives on the stigma. So SCR will tightly bind to SRK, leading to phosphorylation via MLPK and activation of ARC1. ARC1 will inhibit the XO70A1 and target it for ubiquitin degradation. XO70A1 when degraded will not be able to trigger pollen germination and tube elongation. While when we do not have self pollen, then another player THL1 1 by 2 inhibits the phosphorylation events in the receptor SCR receptor, which leads free XO70A1 not marked by the ARC1 for degradation. Now XO70A1 can cause pollen germination and tube elongation. So that proves our fourth statement is the false statement, because tight binding of successful receptor ligand complex SRK SCR leads to inhibition of pollen tube germination in brassica. That is why option 4 is the correct answer. I hope it is clear to the students. Just let me know in the chat box with a yes or no. Good. Great. Okay. Moving on with the next question. Question number 9. Which of the options given below are correctly matched? This is also a match the following based question. Which of the options are correctly matched? GABA gradient, pollen tube germination. perfect flower, hibiscus. Apomixis, no fertilization required. Sus1, dicer or solog. So these are the matched statements, matched pairs, and you have to answer which of these are correctly matched. Option A, 1 and 2 only. Option B, 1, 2 and 3 only. Option C, 3 and 4 only. Option D, 1, 2, 3 and 4. All of them. Now you have to see which of these pairs are not matched correctly. Then you can figure out the rest saying these match correctly. So GABA gradient, pollen tube germination, perfect flower, hibiscus, apomixis, no fertilization required, sus1, dicer homolog. Please let me know what do you think are the correct answers in the chat box. Don't feel shy. If your answer is wrong, no issues. We'll reach to the right answer and we'll understand the concept about it so that whenever you face such a question, you will be confidently able to attend that. So let's take a minute and collect some answers from the students. Okay, it's time for me to reveal the correct answer. The correct answer to this question, match the following question, is option D, 1, 2, 3 and 4. All four of these are correctly matched. GABA gradient and pollen tube germination, perfect flower, hibiscus, apomixis, no fertilization required, sus1, dicer ortholog. Okay, now let us study a little bit about all these matches. Here on my left, we see that the GABA gradient, GABA stands for gamma amino butyric acid. GABA is a neurotransmitter in humans and in other mammals like mice. So we have GABAergic neurons which act as inhibitory signals and glutamate neurons which act as excitatory signals. Let's not discuss that, but I was just letting you know. So in a plant system, GABA gradient promotes the pollen tube germination. The GABA gradient guides the pollen tube to germinate inside the style through the stigma and reach the ovary. So on the stigma, which is the receptacle, female receptacle, receiving place for pollen grains, the pollen will land, the compatibility check will happen in the style, and if they are compatible, they are not self-incompatible, GABA gradient will guide them through the septum and funiculus. Septum is the septum and funiculus labeled here mentions the direction of the ovule, and the transmitting tract is through which the pollen forms the pollen tube, transporting the nucleus, tube nucleus, and the gamete nucleus, which will fuse with the female gamete. So we have an ovule here, the micropylar end through which the pollen tube has to enter and fuse with the egg cell. So remember, at the micropylar end we have two synergids and an egg cell. We have polar nuclei in the center, and at the chalazal end we have antipodal cells. So GABA gradient promotes the pollen tube growth through the style and guides them to the micropyle. And the next statement was hibiscus as a perfect flower. So what is a perfect flower? Flower that possess both stamen and carpels are perfect flowers. So both stamen and carpel on the same flower. Monoecious plants, we have different categories. We have perfect flowers with both stamen and carpel on the same flower. These are hermaphrodites, as well as we have carpellate and staminate flowers with solely carpel or solely stamen varieties. But hibiscus is a flower. If you remember seeing a hibiscus plant, you have those red single single to multiple red stalks coming out from the center with yellow anthers. So we have anthers and filaments, stamen constituting the stamen, and single stigma, style, and ovary, which is the female gynosium part. The multiple anthers and filaments, the androecium part, and we have petal as well as sepal, and the ovary houses the ovules, which will form the future seed of the plant. So hibiscus flowers possess both stamen and carpels, which makes it a perfect flower. Now let us study about a little bit for apomixis. Apomixis is referred to as the process of formation of plant from a seed without fertilization or normal sexual reproduction. So generally plants do reproduce sexually and produce their seeds and fruit and propagate, but there are many cases in which other routes of asexual reproduction takes place, and one such is called apomixis, process of formation of plant without normal sexual division or fertilization, no mixing. And they are also directly developed from the diploid eggs present in the embryo sac. Sometimes what happens, the embryo sac, which harbors uh, which should harbor the haploid ovule. So we have normal sexual embryo sac formation. We have megaspore mother cell 2n, then undergoes meiotic divisions to form functional megaspore. 3 of them degenerate, and then this functional megaspore will undergo mitosis to form the 8-celled 7 nuclei stage, where the egg is haploid and the central cell is the 2n. But what happens in apomixis? We have a megaspore mother cell 2n. There is no meiosis, but the megaspore mother cell persists, so there is no reduction division. So now the inside of the embryo sac is 2n or diploid. Then if this can develop into a central cell undergo mitosis, similar process to form these 7-celled stage, 8 nuclei stage. We have 3 antipodals, central cell with 4n, because since one of the mother cells is 2n, two such nuclei fuse to produce a 4n nucleus, and egg cell is diploid. Since there is no, the egg cell that is produced is from the mitosis of the diploid nucleus. So we have 2n egg cell. Now this can develop into directly into a diploid individual without even needing the pollen for fertilization. So such reproduction methods are termed as apomixis. And on the right hand side is about an enzyme sus1, which is the ribonuclease having homology with Drosophila player dicer. That's why they are orthologs. So orthologs are homology sharing proteins in different species. So we have sus1 and dicer as orthologs in in plants and in Drosophila respectively. So this clears all the four match the following options were correct, where we had GABA gradient promotes pollen tube germination, hibiscus is a perfect flower, in apomixis we do not require fertilization, and sus1 dicer are orthologs. I hope this is clear to the students. Let me know in the chat box with a quick yes or no. Yes, great. Moving on to the next question. Question number 10. This is the fill in the blanks question, like we had in question number 5 or 6. We have 4 fill in the blanks. Unlike dashed do not have continuous contact with mother throughout development. Additionally, the dash is embedded in maternal tissue which makes the study of maternal effect gene difficult. However, the zygote, the dashed zygote develops independently of other tissues. So there are four fill in the blanks, and here the context is maternal effect genes and accessibility of the zygote, developmental characteristics. Option A, plants, animals, embryo sac, maize. Option B, animals, plants, microsporangium, brown algae. Option C, plants, animals, pollen grain, maize. And option D, animals, plants, embryo sac, brown alga. Go through the statement very carefully and try to recall Professor Subramaniam's lecture. He has mentioned it clearly. You might be able to recall that. Go through the question and try to answer the correct option. Let us wait for a minute to collect some answers. Then I'll reveal the correct answer. Okay, it's time for me to, to reveal the correct answer. The correct answer to this question is option D, animals, plants, embryo sac, brown alga. These are the correct fill in the blanks. Unlike animals, plants do not have continuous contact with the mother throughout development. This is true because the megaspore, the megaspore mother cell is a cell which divides into four via meiosis and produces the egg cell or the egg embryo sac, which is now not connected with the mother throughout the development. So it stays as a haploid. As well as in animals, if you see on the contrast, we have placental animals staying in the contact with mother. So and there are organisms like Drosophila which is not placental. The mother lays down maternal effect genes inside the egg cell. Additionally, the dash is embedded in maternal tissue. The embryo sac is embedded in maternal tissue, which makes the study of maternal effect gene difficult. So it is embedded deep within the maternal tissue, so it's less accessible. As in comparison to Drosophila, where we have the egg laid and you can dissect it and you can study it. Defects in development can be studied by the cuticle preparation. So such facilities are not available in plant development, where the embryo sac is embedded deep inside maternal tissue, which makes study of maternal effect gene very difficult. However, the brown algae zygote develops independently of other tissues. So very primitive organisms like brown algae, the zygote develops independently of other tissues. So here is an excerpt from the lectures that Professor Subramaniam is directly teaching. Angiosperm embryos are deeply embedded in multiple layers of tissue, complicating investigations of embryogenesis. So that is why angiosperm embryogenesis is very difficult to investigate, because it is embedded in deep layers of tissue. So within the ovule, within the ovary we have the embryo sac, then we have fertilized ovules growing. So multiple layers of tissue to access the embryo ovule to understand the embryogenesis. Therefore, scientists use brown algae, which is a primitive plant where the zygote is independent of tissues. 8 hours after fertilization, the brown algae look like this. They have the secretory vesicles. They form the rhizoid and thallus to separate distinctions. 25 hours after fertilization, we can see the cell plate formation. So this is used as a model to study the embryogenesis brown algae, which is because the zygote is fairly accessible and independent of other tissues. That's why the studies are mostly made in brown algae. But embryo sac, deeply embedded in maternal tissue, makes the study of maternal effect gene very difficult. I hope you are able to recall this excerpt from lecture 10, lecture week 9 of Professor Subramaniam, and it is clear. Let me know with a yes or no, and we'll move forward. So we have two more questions to go, 2 to 3 final questions, and which will end the session. Since we had a multiple fill in the blanks integer question with question number 567, we have more than 10 questions here, since that would be considered as a single question. So we have two more questions and we'll answer them correctly, understand the logic behind it, and that will be the end of the session. Okay, great. Let's move on to the next question. Question number 11. Choose the correct option. Regulation of planes of cell division and cell expansion are central to morphogenesis in plants. Regulation of plane of cell division and cell expansion are central to morphogenesis in plants. Statement B, plant cells do not migrate. The only exception is the migration of generative cell during pollen tube germination. Statement B talks about plant cell migration. An exception to that is generative cell during pollen tube germination. And statement A talks about plane of cell division and expansion and morphogenesis in plants. Now, which of the option is correct? Option 1. Statements 1 and 2 are true, and statement 2 is the correct explanation of statement 1. Statement 1 and 2 are true, but statement 2 is not the correct explanation of the statement 1. Statement 1 is true, statement 2 is false. Statement 1 is false, statement 2 is true. Statement 1 and 2 both false. So these are different combinations of options you have before you. Read through the statements carefully and use your logic to reach the correct answer. Whether statement A is correct, B is correct, and whether B is a correct explanation of statement A, or one of them is false or both are false. Okay, let's take a minute or two to collect some answers and then we'll move forward. Okay, it's time for me to reveal the correct answer to this question. The correct answer to this question is option A. Statements 1 and 2 are true, and statement 2 is the correct explanation of statement 1. This is the correct answer to this question. Regulation of of cell division and cell expansion are central to morphogenesis in plants. Why? Because plant cells do not migrate. That's why cell division and cell expansion, plane of cell division and plane of cell expansion become so important for regulating morphogenesis. Also, one exception to migration in plant cells is generative cell during the pollen tube germination. So here is a representation of the different cells and nucleus, the how the pollen grain looks like. Germination of a pollen grain and formation of male gametophyte in an angiosperm. This is a pollen grain. The outer covering is called exine. The inner layer is called the intine. It has a grainy cytoplasm with a nucleus. And here is the germ pore. So the germ pore will form the future route of pollen tube growth. Now what happens inside a pollen grain? The single nucleus divides, the single cell divides into two distinctive cells, tube or vegetative cell and a generative cell. So the nucleus, tube cell, so two parts of the pollen grain, tube cell and a sperm cell. Sperm cell is called generative cell, tube cell is called vegetative cell. The tube nucleus is this, and we have a callose wall against the sperm nucleus, and the germ pore where the exine and intine both meet together so as to create a thinner opening. And we have different vacuoles for providing food storage and space. So what happens when there is successful pollination compatibility within the pistil of the female? This germ pore will open up. The tube nucleus will lead the tube nucleus will leave the pollen tube, followed by two male gametes with this callose wall or callose plugs and cytoplasmic seed. So tube nucleus will germinate, will move inside, and followed by two male gametes of the generative cell. So that is how the migration. This is the only place where we see cellular migration in plants. Generative cell during the pollen tube germination. And as we see in radial patterning and axial patterning in the globular stage and heart stage, why is the plane of cell division very required? We can easily spot it here because in absence of cell migration, we do not have cell migration here. The division in which the plane in which the cell divides can determine the amount of morphogen the cell is receiving. The procambium is source of many morphogens. Now if the cell divides in a plane where we have a cell closer to the procambium and one closer to the protoderm, so the one closer to the protoderm will have a diffused or lesser gradient of a morphogen in compared to the cell closer to the uh, center of the procambium. In absence of cell migration, regulation of the planes of cell division and cell expansion are central to morphogenesis. So this is how we form the gradient for determining the root and shoot axis. We have procambium, cell division, cells divide longitudinally, also transversely to create this kind of structure with varied degree of morphogen gradients. Thereby, plants use the altered plane of divisions as well as cell expansion to create morphogenic gradients, which are central to morphogenesis. I hope this is clear. Please let me know with a yes or no, and we'll move on to the final question. Okay, great. Now the final question, which of the following statements are false with respect to plant development? Which of the following statements are false? Option A, suspensors can form embryo-like structures. Option B, radial patterning is visible in the globular stage. Option C, axial patterning is visible in the globular stage. Option D, PID protein kinase is responsible for PIN1 localization. So these are the four statements and you have to tell me which of the following statements are false with respect to plant development. Let's take a minute and collect some answers. A minute later, I'll reveal the correct answer. Okay, it's time for me to reveal the correct answer to this question. The correct answer to this question is option C. Axial patterning is visible in the globular stage is incorrect, is false, whereas radial patterning is visible in the globular stage. So this will be the correct answer to the question number 12. So regarding statement 1, 2 and 4, we will see yes, the suspensor cells can become the embryo. They have full potential to become the embryo, but signals from the embryo suppress embryonic development in the suspensor cells. So there are signals which inhibit suspensor fate, suspensor to embryonic fate, and suspensors send supportive signals. Signals from the suspensor cells promote embryonic development. That's why the suspensor remains as a suspensor and the other regions develop into hypocotyle, cotyledon and other forms of the embryo. And in the globular stage, radial patterning can be easily seen where we have procambium, ground layer, protoderm. Radial patterning in a circular direction, we can see some patterning. Not in the top to bottom, which is the axial patterning, which is not seen in the globular stage. And for the fourth statement, PID is the protein kinase which is responsible for PIN1 localization. And PIN1 is a protein which helps in auxin transport, auxin efflux carriers. So we have a wild type concentration of PIN1 auxin efflux carriers is here. Thereby we have auxin induced gene expression. So since the carriers also called PIN1 is expressed here. So all these cells now will have efficient auxin transport. Thereby we have auxin induced gene expression. Similarly, if we overexpress PID in these domains, if we overexpress PID, no PIN1 auxin efflux carrier here, we remove it to these domains, then PIN1 will be concentrated here due to PID overexpression and the auxin signal will be concentrated here. So auxin induced gene expression will be seen here. So this proves that statement 4, statement 1 and 2 are all correct, whereas the false statement is axial patterning is visible in the globular stage is a false statement. It is radial patterning that is visible in the globular stage. With this, we have come to the end of the problem solving session. I hope you got to learn something new today or revise your old topics. I'll stop sharing my presentation. Yeah, with this we have come to the end of practice problem session for week 9. Next Saturday we'll be solving practice problem weeks at 10, and 3 more classes to go to finish all the practice problem sets. And hope you guys attend all these TS sessions and derive something fruitful out of these discussions and the explanations. If you are not able to attend, please watch the recorded lectures as well as the TA sessions. Practice sessions are recorded, so you'll find the YouTube link from the NPTEL team. And keep practicing your assignments and revising your concepts. And see you soon next Saturday. I'll be ending this call now. Thank you for joining.